To catch prey in total darkness, a barn owl relies entirely on hearing. But intercepting a moving target introduces a significant biological problem. First, there is the sensory delay. The time required for sound waves to reach the ears, convert to electrical impulses, and travel through the nervous system to the brain takes roughly 8 milliseconds. Then there is the motor delay. Once the brain decides to strike, executing the physical movement, turning the head and locking the talons, takes about another 100 milliseconds. This diagram plots the timeline of a moving target. Combine that 8 millisecond sensory lag with the 100 millisecond motor execution lag, and you get a mathematical gap between where the prey is and where the owl's brain perceives it to be. If the owl aims at the exact location its auditory system currently reports, it will strike the space the prey has already vacated. To succeed, the animal must target a point in space the prey hasn't reached yet. The barn owl tracks these targets using the optic tectum, a midbrain structure that organizes auditory cues into a topographic map of space. For decades, the consensus held that these sensory maps were static. If a sound comes from 20 degrees to the left, a specific fixed coordinate on the neural grid fires. A 2006 study published in Nature Neuroscience by researchers Ilana Witten, Joseph Bergen, and Eric Knudsen challenged this static model. Their electrophysiological recordings revealed that the map's spatial sensitivity physically warps in the direction of an approaching sound. By bending its spatial coordinates in real time, the optic tectum functions as a predictive engine. It cancels out the biological delays of the nervous system, allowing the animal to strike the exact future location of the prey. Previous studies had tried to measure auditory motion by sweeping a single continuous sound wave across a neuron's receptive field. This diagram illustrates the flaw. A neuron's receptive field naturally sharpens shortly after a sound begins. If you sweep a continuous sound across it, it is impossible to separate onset sharpening from the actual spatial shift. To isolate the true effects of motion, researchers had to measure the entire receptive field at identical, locked time points following the sound's onset. To achieve this, the team simulated motion using dichotic sounds played directly into earphones. They created virtual movement across the horizontal plane by adjusting interaural time differences, the microsecond delay between sounds reaching each ear. This animation shows their protocol. Instead of a single continuous sweep, they presented a randomly interleaved series of short sweeps originating from evenly spaced, staggered starting points. By compiling the neural responses to all these distinct sweeps, they could build a snapshot of the entire receptive field at an exact millisecond. Any shift observed under these conditions would be a measurement of spatial prediction, free from the confounding variables of onset latency. We can see exactly how the neurons responded to this simulated motion by analyzing the resulting spatial tuning data. This contour plot shows the neural response to a moving sound. The black line is the static baseline. Initially, the receptive field is centered. By 300 milliseconds, the peak sensitivity physically drifts, shifting by 14 microseconds in the final window. The direction of this drift is precise. For a sound moving left, the neurons shift their tuning toward the right. For rightward motion, they shift toward left ear leading time differences. The neurons abandon their fixed coordinates. They actively adjust their peak sensitivity to map the exact location the sound is about to enter. For this to be a predictive mechanism rather than a fixed reflex, the shift has to scale mathematically. If the target moves faster, the neural map should warp further to compensate. The researchers tested this by varying the speed of the simulated motion, presenting virtual sweeps moving at 5, 20, 40, and 80 microseconds per second. This graph tracks the predictive shift over time. As the stimulus velocity increases, the shift increases in proportion to that speed. The highest velocity, represented by the thickest line, drives the most extreme spatial shift. To translate this spatial shift into a unit of time, the researchers divided the shift's magnitude by the moving stimulus's speed. This mathematical calculation yields what we call the predictive time lead. This scatter plot shows the time lead across all tested speeds. Regardless of how fast the sound moves, the system consistently calculates a predictive lead of roughly 100 milliseconds. That 100 milliseconds aligns with the duration required for a barn owl to physically execute a saccadic head turn to acquire a target. The neural shift scales to cancel out the bird's mechanical limitations. The map warps by the mathematical equivalent of the owl's physical lag. Calculating a dynamic, speed-dependent trajectory using static anatomical hardware requires a specific computational mechanism. This model relies on negative feedback. A central excitatory zone is flanked by an inhibitory surround. Excitation in the center suppresses future gain, dragging the lagging edge forward. Conversely, suppression in the surround increases future gain, advancing the leading edge to meet the target. By pairing adaptation with lateral inhibition, a static network is transformed into a predictive motion tracker. To confirm this mathematical model, the researchers had to test if these exact feedback mechanisms were physically occurring in the living optic tectum. They designed a biological test, playing a static 250 millisecond priming sound in either the excitatory center or the inhibitory surround, before immediately playing a second test stimulus. This chart plots neural responsiveness following an excitatory priming sound. Compared to the baseline, the dashed curve is severely flattened. Neurons physically decreased sensitivity, confirming response adaptation. Conversely, this third curve shows reaction after an inhibitory sound. The neurons amplified responsiveness, proving post-inhibitory rebound. The computational model aligns with the biological reality. These basic cellular behaviors drive the spatial predictions observed in the math. Because space is mapped topographically in the optic tectum, a physical location on the brain tissue directly corresponds to a location in the external world. This animation illustrates the macroscale implication of those receptive field shifts. As a sound moves, the active neural population separates from the static auditory coordinate, the black oval, and slides across the grid, forming the red oval. The location of that neural activity serves as the encoding for a motor command. The brain instructs the body to aim at the red oval, the future location of the prey, rather than the black oval representing the present. The result is a predator launching an attack into empty space, closing its talons just as the target arrives. Sensory maps in the central nervous system function as fluid predictive topologies. They warp sensory input to ensure our physical actions meet the future exactly on